If you've been following the news for the past decade, you may have heard of superbugs, or bacteria that are stubbornly resistant to antibiotic treatments. These sneaky bacteria have evolved mechanisms that allow them to cancel the effects of antibiotics. Since they cannot be inhibited by conventional antibiotics, antibiotic-resistant bacteria pose one of the most pressing issues that our generation faces. Antibiotic-resistant bacteria sure sound alarming, but how did they become resistant in the first place? And most importantly, why should you care about antibiotic-resistant bacteria? In this short video, we will explain what antibiotic resistance is, how bacteria can acquire antibiotic resistance, and how you can help slow the development of these nasty superbugs. What is antibiotic resistance anyway? Antibiotics are compounds that inhibit the growth and survival of bacteria by targeting bacterial functions that are essential for life. Depending on what cellular function they attack, antibiotics can be effective against broad groups of bacteria or only very specific bacterial species. For example, penicillin is a common antibiotic which prevents bacteria from forming cell walls. Therefore, penicillin is very effective against cells with a thick cell wall, which is important to the bacterium. However, bacteria have developed methods of avoiding the effects of antibiotics or modifying antibiotic compounds. In case of penicillin, some bacteria have evolved DNA encoding for proteins which break the structure of penicillin, rendering the compound ineffective. But how do bacteria become resistant? When a bacterium duplicates in a process known as binary fission, it copies its DNA. This process can be rapid. Some species of bacteria can duplicate every 20 to 30 minutes in conditions with adequate nutrients, available water, and ideal temperatures. Since DNA replication occurs so often, mistakes, called mutations, are likely to occur. Mutations are defined as changes in the DNA sequence of an organism compared to its ancestor. Though the word mutation is commonly associated with harmful cancers in humans, mutations can actually be beneficial for bacteria. However, what is helpful for bacteria is not always helpful for humans. Mutations that cause antibiotic resistance make infections more difficult to treat. Another means of antibiotic resistance development involves the acquisition of DNA from other microorganisms that are already resistant. This process is called horizontal gene transfer. It is the movement of genetic material between bacteria and other players in their environment. In a way, Spider-Man acquiring powers from a spider is like bacteria acquiring antibiotic resistance genes by horizontal gene transfer, which then give them the power to resist antibiotics. Antibiotic resistance genes can be acquired through 3 types of horizontal gene transfer. If you'd like to learn more about the process of horizontal gene transfer, check the links in the description. While antibiotics are helpful for stemming the growth of harmful bacteria, the overuse of these antibiotics creates a need for bacteria to evolve to survive in the environment. This phenomenon is called selective pressure. Ultimately, antibiotic resistance comes down to a survival of the fittest. Most vulnerable bacteria of a species will die when subjected to a sufficient concentration of an antibiotic. However, the bacteria of this species which have a corresponding antibiotic resistance mutation or gene will survive. The surviving resistant bacteria will multiply, and a new generation of antibiotic-resistant bacteria is created. Antibiotic overuse and bacterial antibiotic resistance are serious issues that can be detrimental if not managed well. Luckily, we can minimize this threat by following guidelines that have been recommended by experts. First and foremost, reduce the chances of bacterial infections by maintaining healthy hygiene habits and prepare and store your food following food safety guidelines. Next, vaccinate yourself and your children, as bacterial vaccines can prevent corresponding bacterial infections, and antiviral vaccines can reduce the number of viral infections that are often improperly treated with antibiotics. Preventing infections through proper hygiene, food safety, and vaccination decreases the total number of infections, therefore reducing the frequency of antibiotic use or sometimes misuse. If you do get sick, consult your doctor as to whether you need antibiotics. Don't pressure your doctor into prescribing you medication. When we have a cold, it is most likely caused by a virus, so antibiotics will not help us. When we are prescribed antibiotics, we should take the full required dose as prescribed by a doctor, even if we are starting to feel better. After all, feeling better doesn't mean we are better. Sometimes a small population of persistent bacteria can cause reinfection if it's not completely eradicated. When we complete our prescribed course of treatment, we should not save antibiotics for future infections. Antibiotics lose potency over time, so using leftover antibiotics for future illnesses may not help. It is also not recommended to use the antibiotics to treat another illness, as not all antibiotics work against all bacteria. However, some antibiotics affect many species of bacteria. This means that beneficial bacteria in our digestive tract, known as our gut microbiome, are harmed and can lead to digestive issues. Most importantly, we should not share our antibiotic prescription with anyone else. Antibiotic misuse can create selective pressure and encourage the growth of antibiotic-resistant bacteria. We don't want to become an incubator for resistant bacteria. By following these recommendations and keeping up to date with experts in the field, hopefully we, our friends, our family, and not harmful bacteria can remain safe for generations. If you'd like to learn more about mechanisms of antibiotic resistance, horizontal gene transfer, or other concepts mentioned in this video, check the description for more information.